Let's remember our board game friend. Okay, so he approached you with the problem. What was the first thing we asked him about?
Sample size.
Sample size, power, right? And then what was the second thing we asked? No, we haven't asked him about stratification yet. Soon we will. Multiple hypothesis testing, right? And then so let's say that our board game friend, he does this, he's worked out the deals, he has a huge board game sample, and he does his discovery, and then he comes to you and he's so depressed because he's like, I tried to replicate this, and none of my things replicated. They're all way smaller than I saw in the discovery. What do you tell him? Winner's curse. Okay, so these are three things. So I'm focusing on these things because if you come to me and you say, I have this great idea, I'm going to ask you the same three things as well. So you may as well ask yourself before you come to me too, right? Or Dan or David, we're all going to ask you these things. These are I think some of the big fundamental problems that you need to be thinking hard about as you're doing your own research. And if you deal with those things, then I think that's a really big first important step in making sure that the research you're doing is good. Okay, great. So next we're going to talk about stratification. We've been talking about this already all week long, so I think some of you have an idea of what it is in principle, but I'd like to dig into it a little deeper and understand where stratification comes from and what we can do about it. And so this is the example that we gave here, the chopstick gene. This could also be like a borscht gene or haggis, or I don't know. But yeah, so the idea is if we do this GWAS and we say we're going to do this GWAS on chopstick use, and we find all of these hits, we for sure will find a ton of hits if we have a sample that includes both Asians and non-Asians. But do we think that these SNPs actually have a biological interpretation? No, because we think that probably these SNPs are just telling us about a person's ancestry and not about some process that starts with this SNP and then causally leads us to whether or not they can use their fingers with chopsticks. Okay, so let's be a lot more precise than that now. So I'm gonna start with this model here. So we have some phenotype Y that we have measured on a bunch of individuals. We have these X's are genotypes. I'm going to assume that these genotypes are standardized to have mean 0 and variance 1, just to make our life a little easier. And so each one of these genotypes is associated with the phenotype with a coefficient beta. So that beta could be 0, in which case I guess it's not associated, but there is an association, an effect size coefficient associated with each SNP. What I'm going to assume here as well is that there's some additional fixed effect that is a group level fixed effect. So it's something that's correlated with a person's ancestry that's affecting the phenotype measure, but it's affecting it independent of the person's genes. So in the case of our chopsticks, this would just be like if a person's Asian, then in Asia they use chopsticks, and so it's an environmental thing that causes them to use chopsticks more frequently, but it doesn't have anything to do with their genes. And so that's the type of things that live in this G term. And so we're like, I'm from Utah, and people from Utah eat a lot of green Jell-O. And so if we wanted to do a GWAS on green Jell-O consumption, you'd maybe want to have that in G as well.
Is that true?
That people eat green Jell-O in Utah?
Yeah.
Yes. I think we're the #1 Jell-O consumers in the United States. Also ice cream sometimes, but I heard that California took that away from us recently. Okay, so and then over here we have some residual. That's kind of everything else that affects our phenotype that is uncorrelated with both these group differences and with these genetic differences. That's got genetic variation, right? So I'm going to assume that these X's can be correlated with each other due to things like LD or a person's group. We're going to talk about what that means on the next slide. But I'm also going to assume that this group difference variable can also be correlated with X. Well, there's lots of correlation that's possible here. Let's talk about why we might have first correlation between the SNPs. So the first thing is linkage disequilibrium. We talked about that a bunch. When you have two SNPs that are close to each other on a chromosome, they're often, they're almost always going to be inherited together. And so as a result, there's going to be some correlation that arises just due to the fact that they're close to each other. If we had SNPs that are on different chromosomes, because they're inherited separately through like different random like coin toss type processes, then those tend to be uncorrelated due to linkage disequilibrium. Within a SNP, the closer it is, because the reason that things on the same chromosome, we don't inherit whole chromosomes at once because of these crossover events that we talked about earlier in the week. And so if they're far away, it's more likely that there's a crossover event between the two SNPs. And so that's why we can think of SNPs that are far away from each other as being less likely to be correlated with each other. And so that's one source of correlation. It has entirely to do with these biological processes. And it's nice because we can sort of think of these LD things as being like this local phenomenon. The other reason that SNPs might be correlated with each other is due to population structure. So let's say that there was kind of one big group, and there was random mating in this big group, and so we know that we'd be in Hardy-Weinberg equilibrium. The allele frequencies are going to be the same. I think that you did a problem showing why that is in problem set 1. But let's say that there's like plate tectonics, and so now these groups are separated and they can't get to each other. But within each group, there's still random mating. Now, just over time, these allele frequencies just may shift due to chance. It's a bit of kind of a random walk-like process. And so kind of group on continent A, over time, maybe the allele frequency at one of their SNPs is going to go up a little bit. On continent B, the allele frequency may go down a little bit. And all of a sudden, if we look at a person from continent A, or if we just draw a random person, we don't know what continent they're from, but you tell me what their genotype is at this particular marker, if I see that they have one or two copies of this marker, then it's going to make me think that they're more likely in this group where that marker is more frequent, right? Because in this group, it's not very frequent, and so if I see that marker there, then it's like, oh, they're probably not from here. So let's say that there are two such SNPs that have drifted in the same way. So I can look at the one SNP and I say, oh, this SNP is telling me that this person is from this group here. And so it's going to be correlated with the person's group. This SNP is also correlated with the person's group because of these changes in allele frequency that have occurred over time due to non-random mating. And therefore, these two SNPs are going to be correlated with each other. Now, this effect that I'm describing can happen anywhere in the genome, right? So you could have a SNP that's on one chromosome, and it's become more frequently in one of the groups. And you can have a SNP on another chromosome, and it's become more frequent in that group. And all of a sudden, we have two SNPs that are correlated with each other that are on totally different chromosomes. And so this is one of the reasons that population structure can induce this correlation. A nice little result is that you can actually separate the covariance. So the covariance of a pair of SNPs, you can decompose it into two pieces. It's actually additive. And so we have correlation due to things like LD, and we have correlation due to these kind of population things that we're talking about. And so I'm going to use that to talk about GWAS coefficients now. Mm-hmm.
So is it possible in practice to decompose the variation or the covariation between the SNPs into the covariation arises from LD and that arises from population structure?
You would need to have something that allows you to kind of separate that out. Maybe like a within family group. I mean, like, it seems like one could, but you'd have to figure out some way to be able to decompose this.
Doesn't the separate chromosome thing do that for you?
That's true. So if you saw that two SNPs were on different chromosomes and you see any correlation, then you must know that it's from this group here. The problem is that if they're on the same chromosome, you don't really know what this is. It has to do with kind of how much crossing over you expect to see in different parts of the genome. So it's a little more tricky if you want to actually decompose it between two SNPs on the same chromosome. I mean, I think that there are people who've tried to do it, and I think that when I've seen it done, it's been done with kind of within family. I mean, they don't directly try to do it, but they get at that kind of thing. Mm-hmm. Joey?
So is every marker in the genome, does it vary by ethnic background, or is it just a subset of all markers?
I would assume that if the groups have been separated for long enough, then, I mean, it might the differences might be very, very small. But the process that I described about how they might separate, like it's a random, you know, and it's like this random walk thing, and so it should vary a little bit, but maybe not enough to be practical. There are going to be some SNPs that vary a lot, and those are going to be the ones that cause a lot of problems.
Could you get down to like a manageable number of SNPs that vary very little across races and ethnic backgrounds and put them together?
Yeah, I mean, it's tricky to identify them, though, right? We don't have really precise estimates of the covariates of each SNP to begin with. And then if you're trying to look for like small differences, the amount of noise you have in your estimates of the LD between SNPs, or I guess I should say covariance between SNPs, because is small relative to the amount of error that we have in our estimates. Bernie.
So yeah, so it could be drift, but couldn't it also be selection?
Yeah, so I didn't want to dig into that too much, but selection could do it too. So like, let's say that there's, you know, people on continent A love tall people, and people on continent B love short people. Then SNPs that make you taller are going to be a lot more prevalent on continent. They're going to, you know, it's going to be pushed up to be a lot more common in this group than for this group. And so yeah, selection could do it too. Selection is just a little bit more messy. So I was trying to give the clean example.
It seems like this structure of linkage, this equilibrium, and its local nature should make it possible to get these two variances. And the reason it could maybe be worth trying to do that, if the literature hasn't, is because you want to know how much total variance there is because of stratification problems.
So that's what you're talking about is LD score regression. We're going to talk about that all day tomorrow. LD score regression does take this
No, but can't you do something related to that that also tells you the total amount of covariance between SNPs that's not about LD, and therefore tells you how much stratification you need to worry about?
Yeah. They'll do that with LD score regression.
This question of how many PCs you could get, because you could say there's this total amount of variance. We want to absorb almost all of this variance, and we're not there yet.
Yeah. So this will be a consequence of LD score regression. We'll talk all about that. But you're exactly right. This is we can use that information to see kind of how much variation is kind of left over after just accounting for what we think is due to LD.
And when you do that, then what does it say about how you're doing with 15 PCs or something relative to the total amount of the stratification?
Yeah, so it's very trait specific. But in general, it seems like the amount of variance due to things like stratification is limited. What's the estimation for like EA? It's something maybe like 8% is the implied, you know, so there's the amount of variation that we see in our estimates due to stratification is perhaps 8% of the total variance in that ballpark. But it's also a little bit noisy because you have to, Those
at that 8%, how much does your 15 PCs get?
You know, I don't know that we've done this carefully, like saying what's the implied variation with different levels of PC. That might be interesting, but I don't think I've seen it.
If there's any way to separately, I mean, it's kind of like what you do with the heritability, but if there's a separate way to estimate how much stratification variance there is in the sample, and then you compare that to how much stratification variance you've captured with the PCs, now you've got a better way of talking about how many PCs you need.
Yeah, that's a reasonable. Yeah, I mean, it's maybe worth looking at. Mm-hmm.
So if we don't actually think that G1s are tagging the causal SNP, like have we found evidence because of this population in different populations that like there might be a significant SNP that's true for Africans and Europeans, but like the sign's different? Like it's actually sort of tagging like a different sort of polymorphic or a different sort of mutation. Does that question make sense?
No, say it again, please.
So like, let's just say that there is a SNP that varies in both European and like African populations, but because like actually what it's capturing, where it's not actually that variation that's important, it's like the things that are around it. And so on average in European populations, the things that are around it is pretty different than on average the things that are around it in African populations. And so like maybe there's like a deleterious mutation in African populations that are around this SNP, but like a positive mutation in European populations, or vice versa. But just like the sign is different for a given SNP in different populations. Does that make sense?
Yeah, so I mean, I think that this is getting back at this idea of fine mapping that we talked about before. And so it is, you can try to use differences in LD structure to try to identify kind of which SNPs are causal and what they're doing just by looking at the differences. But you have to believe, to do something like that the causal SNP is the same between these populations, where it sounds like you're saying, what if it's actually, so then there's kind of two dimensions of differences, and so that's hard to deal with, right? Because we, in order to identify the causal SNP, if we have two populations, we're saying we're holding the causal SNP constant and kind of triangulating. Whereas if you're both allowing the effect size to change and LD to change, you'd need something else that you can hold constant to like nab that, right?
I'm just sort of interested, like, do we have evidence that in different populations the causal SNP is different for a significant loci?
I mean, it's going to be trait specific again. I don't know of any. James maybe has. It looks like you have something.
So Matt Keller was a senior author of an interesting paper a few years ago that looked at this for schizophrenia. They calculated the genetic correlation between Europeans and Africans. So Matt Keller, he'll be here shortly. And they found that the genetic correlation, I think, was 0.65 with a larger standard error, but at least that suggests that it's not like just totally different. And also I believe that maybe a few months ago there was a paper along similar lines from maybe the Broad group on bioarchive that looked at some other traits, and I think they found similar results. Maybe that one was Europeans and East Asians, where genetic correlations are largish with maybe not. I think they could statistically reject it as equal to 1, but still they're on the high side.
Okay. Great. So let's talk about stratification then and these different kinds of sources we're talking about. So let's say that I wanted to do, I guess this should probably say expected value of beta GWAS, but a GWAS estimate, when we're doing a GWAS, we only regress the phenotype on a single SNP at a time. And so just by the properties of OLS, the coefficient of that OLS estimate is going to be the covariance between the phenotype and the specific SNP divided by the variance of that SNP. So at the very beginning I said we've standardized our SNPs, so the variance of the SNP is 1, so we're just going to get rid of it. Next what I'm going to do is, a couple slides ago I said, well, y is equal to this thing. So I'm going to plug that, this in, because we've assumed that this is what y is. And now what I'm going to do is, because covariance is a linear operator, I think it's called like bilinear, something else, because it can go in both directions. But we can essentially look at the covariance across each terms of this sum. And so I'm going to pass this covariance of x through. And so now we have the sum over the covariance of SNP I and SNP j due to LD times beta. And then plus the sum of the covariance due to population structure times beta, plus the covariance of the SNP itself and these population fixed effects. Okay, so there's 3 pieces here. So I'm going to try to argue that this piece here is not super concerning. So all of these are kind of sources of what we might think of as omitted variable bias, if you've seen that literature. But we can see what we maybe think we want are these betas conditional on every other SNP. But we're getting this weighted average of a bunch of different betas because we're omitting them in this simple regression. So this term here, why might I say that there's still some interpretation about this term? Like what's going to be the contribution of SNPs far away to the bias in this term? Like SNPs on different chromosomes. Does that make sense what I'm asking? I'm probably not being super clear. So what I mean is, so this LD is correlation due to the LD structure. And so that's only going to be non-zero for SNPs that are close to each other and then 0 otherwise. And so that means that even though this term that we're going to be estimating is not the beta that maybe we're interested in, it's the sum of a bunch of betas for things that are in LD, which means it's the sum of betas for things that are close to each other. And so as a result, we can't interpret GWAS coefficients as the effect of the individual SNP, but we can pretty safely interpret them as the effect of some set of SNPs in an LD region. And so, and then given that we're often going to take this into like a bioanalysis, and we're going to say, well, what genes is this SNP close to? Given that there's maybe a little bit of fuzziness because of this term in the GWAS, and then a little bit of fuzziness in saying like, oh, what gene is this close to, or any kind of follow-up analyses, that's a type of fuzziness that we're okay with. Okay? Over here, though, what's the problem with this term? Why am I less comfortable with the bias in this sum, the second term of the sum? Yeah, we totally lose that interpretation that we had before, because these things can be non-zero throughout the whole genome. And so, if there's stratification, and then you give me a GWAS coefficient, I have no idea what to do with that, right? I mean, all I can say is that, oh, it's associated with this SNP or any SNP that's correlated, which could be any SNP. And so that's not very interesting. Let's talk about this term here. So this term is how the SNP itself is correlated with these kind of non-genetic factors. And so this is also a problematic term. This effect here is what we think of as the chopstick effect, the chopstick gene, because the SNP is going to be correlated with, just things about your ancestry, environmental things that might affect where you're from. And so so kind of these two things together is what I'm thinking about when I'm talking about stratification bias. And so both of these are a result of these differences in mean that are going to exist between these groups. Because there's a difference in the allele frequency in one group and the other, it induces correlation throughout the genome and correlation with these non-genetic effects. This one is, in principle, just kind of as a side note about this guy here, this one shows up as a result of us not controlling for all of the SNPs at once. So in like a theoretical regression, if we could control for every SNP in the genome, this term would go away. We'd be okay. But this term would still remain, and so we still would need to worry about that kind of stratification. And I think that most people are concerned with this last term more than the second one, but the second one still is there. Mm-hmm. Yeah.
Just trying to get a handle on it. So I feel like the sociologists and population scientists use the word stratification differently. And so in the middle, that first, the Popeye J, that's what we would think of as sort of biological population stratification. And then on the right is sort of the social stratification part of it.
Yeah, so I think the stratification in general, when we say that, we mean that there are groups that are separated in some sense, right? And so that's kind of the catch-all term that we use when we're saying oh yeah, this group is different than this group. And so yeah, so I don't think I've heard people use the language that you just used now, but I guess, yeah, you could say that this is due to how different SNPs are stratified across groups. And this is how both the SNPs are stratified and kind of these other, you need both the SNP and the G to be stratified for this bias to show up.
So I guess, maybe this is a better question, is that, so I understand the one on the right is sort of the overlay of this environmental differences and then this population stratification. So the one in the middle, if we didn't have the one on the right at all, would the one in the middle still be a problem?
Just in the sense for this interpretation sense.
We would be confused if we were getting the right spot in the genome.
Yeah, so let's say that there's that really strong smoking gene that Dan talked about earlier. And let's say that there's some population where their allele frequency for that smoking gene is different than in this other group here.
That's actually true. So the bad variant is more common in Europeans.
Yeah. So it's actually, this is a great example that I came up with. Then if we took any other SNP in the genome that was similarly stratified, that SNP is going to pick up this super strong chromosome 15 effect. But we don't necessarily know that, and so it's problematic.
So if we didn't have any of the covariants G and X, we would still derive the right heritability or SNP heritability? Is that true?
Heritability is really hard to talk about when your SNPs are correlated with things in the environment.
So I'm saying if they weren't. I think that's the
Oh, if it's not.
Both of those are bad, right?
Okay, yeah.
Figure out how they're differently bad, and the one on the right, if we just got rid of that.
Okay, yeah. So you're just saying there's only this.
Yeah. So if there was only that, then we wouldn't spots we got for our, like, on the genome were like, exactly right, but we would know that we're estimating the right SNP heritability.
Yeah, I mean, I guess it just depends on how are we estimating. Are you talking about like a twin estimate or?
Yeah, I don't know.
I mean, yeah. So it depends on like what you're doing. I mean, in principle, I think yes. LD score regression would still be. I mean, LD score regression is fine even with that last term in principle, like under assumptions.
Well, the twin estimate is within family, so there's no.
Oh yeah, so that should
no
be fine
issue of
too.
stratification.
But yeah, I mean, this is a good point. You know, you should understand that your GWAS, like, depending on what thing you're doing, like what estimate of heritability you're getting, you should think hard about how stratification is going to be implicated. In fact, any research you do. This is why our fourth question for our board game friend is going to be how is stratification affecting your analyses. Because stratification is such a problem, any analysis that doesn't address it is probably wrong. So you need to make sure that it's that you're addressing it and that you're addressing it carefully. Okay, great. How long am I supposed to go? I guess I'll just
keep going.
I'm just going to keep going. Okay. So so first off, so you know, we maybe have stratification, but we're not sure, right? So maybe the first thing that we'd want to do is say, well, what tests do we have? What are ways that we can estimate how much stratification there is in our data? And there's kind of two. I mean, there's probably lots, but there's two that I'm only actually going to talk about this one. So you can use within family methods. You know, so there's kind of a class of methods using within family data that will allow you to assess if there's stratification. LD score regression also allows you to measure how much stratification there is in your data, and Raymond's going to tell you all about that tomorrow. But I'm going to talk about this quickly. Okay, so if we have family data, so we have twins or siblings or some, you know, people who are related, we could instead estimate this model. So this delta thing is the difference. So what we'll do is we'll take SIB pairs, and we're going to take their outcome, we'll take the difference in their outcome, and we're going to regress that on the difference in their genotype. And so the reason this is okay, let's look back at the original model we started with. For siblings, this G is the same thing. So when we take the difference in the phenotype, this thing disappears. And so kind of like we were saying before, if there was no G, if this G thing wasn't a problem, then we should be okay. And so by taking the difference, we're getting rid of this G entirely. What's additionally nice about this is if we look at differences in SIB pairs of the genotype, we're also going to be removing, we're essentially demeaning, there's no longer differences in mean between our individuals. And so, because you know, you've received your genotype randomly from your parent, it's essentially, once you have your siblings and you look at your difference, it's essentially random assignment between siblings. And so these differences in mean go away, and so these cross chromosome problems that we had are also going to go away because these differences in SNPs aren't predictive of your group anymore, and therefore they're not predictive of each other. So this is nice. We've solved this stratification problem by taking differences in siblings. We can run this regression. And as a result, when we do a GWAS of differences, all we're left with is here, because the covariance due to population has gone away, the covariance with the levels has gone away. And so this is the same as it was before. So we're getting exactly the estimate that we would have gotten had there been no stratification in our kind of cross-population effect. So I've made a couple assumptions here. Let's see, I'm gonna. Oh, well, let me just make this note first. So the reason that we're not just doing this to start off with is because within family samples tend to be very small. You know, we need hundreds of thousands of individuals to detect genes, and our family samples have like, you know, a few thousand people. So we can't. Yeah, maybe. So maybe someday we'll be able to just do everything within family and then it'll be clean, but just currently we can't. But these aren't useless because we can use these within family estimates, which we know are unbiased and have no stratification, and we can compare these estimates to our potentially stratified cross-population effects, right? So let's say that we had some phenotype, and we think that actually the genetic component of you know, there's actually no genetic effect. and that all of the things that we're estimating are just entirely due to stratification. And so this is like what people were telling us when we first started doing EA. They're like, there's no way that this is actually genetic. It's probably just all stratification. How do you feel about that? And so a sign test answers that question. So under the assumption that there is no stratification, or that there's no genetic signal at all, this means that all the betas are 0. And so the within family estimates will, again, this should probably say expected within family. So the expected within family estimate is just going to be 0, which means the sign of the within family estimate is going to be 50% positive, 50% negative. And so as opposed to our estimates here, let's see, these estimates here, which if we've assumed that these are 0, we still have this term here. But we're going to use this fact that the sign of our within family effects are arbitrary, and we can use that to test this assumption that the betas are all 0, because in expectation the signs are only going to match 50% of the time, because in our with family the sign is totally random. And so if we can reject, so if we have a much higher sign concordance, so if our sign concordance is like 90% of the time, then it's really unlikely that that everything is just stratification, that there's no genetic effect at all. And so you can do a sign test like that. This is kind of a weak test though, right? I mean, like, do we actually think that there is no genetic effect at all? Well, and then maybe there's some. And especially now that we're doing this follow-up stuff and people aren't trying to tell us that 100% of our results are just stratification, maybe a sign test is not very appealing, because all we can say is like, oh yeah, there's some genes in here.
Can you explain the sign test again? It was a little fast.
Okay, yeah, sorry. I don't really like sign tests. So the idea is, let's say that we estimate we go within family, and we get a within family beta estimate. So if there's no genetic effect at any SNP, then it means that the mean of that estimate is going to be 0, and it's going to be normally distributed with mean 0. And so because it's normally distributed with mean 0, it means half the time we're going to estimate something positive, and half the time we're going to estimate something negative. In our GWAS, though, we're going to have some effect, and we can't really say anything about what the sign there is going to be, because it's driven entirely by these correlations between the SNPs and this G, the difference in levels. So I'm not going to say anything about the sign. However, because the within family test is independent of our GWAS, and it's just 50-50 on the sign, then if you tell me what the sign is for the GWAS, then there's still just a 50% chance that we happen to be on the same side, and a 50% chance that we happen to be on the other side. And so again, we're under the null. We're saying that every single SNP is like this. So for every single SNP, there's a 50% chance that our within family happens to be on the same side as our GWAS, and a 50% chance that it's on the other.
It seems like there's another potential problem with the sign test, if I'm not making a mistake, but seems like there are evolutionary reasons for minor variants with small effects to often be deleterious. So you've got a deleterious thing. You have things that appear by mutations. They're only a tiny bit bad, so it takes a fair amount of time for them to be pushed against by evolution relative to given that they have a tiny effect. And so this sign test would be one thing if the direction of up were random. If whether your reference was the minor allele or the major allele was totally chosen randomly. But if you had your data so that your minor allele was always a plus, then it's not at all clear to me that it should be a 0 mean thing. So if you do a sign test, it seems like you should try to make sure that the direction that's the minor allele is randomized.
Are you assuming, though, it sounds like you're assuming there's already effect. And we're under the null, we're saying that the effects are 0 to begin with. So like there's no deleterious or positive or anything, because it's all zeros.
Oh, well, okay.
Yeah, so that's the null hypothesis.
Also, the reference alleles are pretty much random.
Yeah, and that's also true. We tend to, Yeah, we can think of the reference alleles, what we're choosing as the reference alleles as random in most cases.
Just to clarify, so you said that under the null, we expect 50% of the time that the within family coefficient sign matches the GWAS coefficient sign. What about under the alternative hypothesis?
Good question, Dan. So under the alternative, so we said this is what our estimate is of of our GWAS, and so under the alternative, this thing is not 0. And so we have this component that lives in the GWAS. We have that same component that lives in the within family. And so because this component should be the same in both of these groups, they're going to tend to have the same sign if there are true genetic effects, because this is capturing the true genetic effects that are all of a sudden non-zero. And so that's why when we're doing a sign test, we're looking to see if the amount of time that these SNPs have the same sign is greater than 50%. Because under the null it's 50, and under the alternative, we don't necessarily know exactly what it is, but we think it's bigger than 50. And so once we look across a lot of SNPs, we may be pretty well powered to be able to look for even small deviations from 50%.
So if you have like loads and loads of SNPs and the deviation is just a bit above 50%, and it'll be significant because you're doing, you have loads of SNPs, what would that really tell you? I mean, how large a deviation you want for you to sort of really believe?
So you'd want first thing to keep in mind if you're doing these sign tests, you need if all of your SNPs are independent. So let's say that we select some subset of SNPs that are all independent, and then we have a variable of whether or not a SNP has a concordant sign. So if they have the same sign, we're going to call that 1. And then we take the sum over all of those things. That's going to be distributed as a binomial. So it's a count of how many matching signs there are. And under the null, it's a binomial with a 50%. So this is fully, we know what a binomial with m SNPs and a probability of 50%. That's fully parameterized. And so we can get a p-value out of that. Now, with the number of SNPs that we tend to have here, very small deviations from 50% tend to be significant. So I'm trying to think, like in the EA paper, I think that we were like 50.2%, but it was strongly significant because there were so many SNPs. I mean, the reason that it still will be pretty close to 50% is even if this term is matching in both of them, there's this estimation error term in the within family estimates that because we have so little within family data, it's very noisy, right? So it's slightly more likely that it's going to be 50% because they share this term, but the noise is orders of magnitude larger than this, and so it's only a tiny bit more likely. But we can detect it if we have thousands of SNPs like we do.
But I think another part of the answer to Bruin's question is you need, to interpret a slightly bigger than 50% sign matching, you need to do the actual calculation of what you would expect if there really is an effect, given the fact that your sibling samples are so small and you have so much noise in those estimates. If it turns out that under the hypothesis that all your effects are real, the expected amount of sign concordance is 50.3%, then 50.3% is a resounding success of the model. If the expectation is 60% and you're getting 50.3%, then you might say, well, we can reject the null that that our estimates are entirely driven by stratification, but it looks like there's a lot of stratification in these results.
But let's say that we did this. Here's a question for you guys. Let's say that we did our GWAS, so now we have our estimates, and then we want to do this power calculation that Dan's talking about. And so we say, okay, under the assumption that our estimates are the right thing, we do this power calculation, and we find that we should get like 55% concordance, but then we just get this like 51%. And you show this means like, oh, we didn't do very well. And I would say, well, maybe you did fine. What did I do wrong in this analysis I described? So let me tell you again. I took my GWAS summary statistics, I used those as the true effect sizes that I'm plugging in, and then I do my power calculation. What did I miss? Winner's curse. Okay, yeah, so again, we're doing these power calculations to figure out how well we're doing relative to what we'd expect. In order to do that, we need to think about winner's curse.
I don't think I understand how you're getting the signs for the siblings. Are you literally just running like a GWAS with 2,000 people, or are you
Yes.
like, oh, that's it?
Yeah, yeah.
Oh, okay, yeah. But so like, we wouldn't expect any of the data to be significant. We're just looking.
Yeah, we're just looking at sign. But if this thing is, kind of large a little bit, then we should be able to detect it with enough SNPs. But again, the null hypothesis again is a super weak hypothesis. So we're saying that every single beta that we're looking at is exactly 0. And so even if we can reject that, that doesn't mean we've learned much. There still could be an enormous amount of stratification.
So let me just add to that. So as Patrick said before, if you had a big enough sibling sample that you could reliably estimate the betas, we would have just done that to begin with.
Yeah.
So the issue here is that we have this small sibling sample, and what we're trying to do is get what information we can out of it about stratification. And the sign test is one way to do that because you're aggregating across these many SNPs and trying to see, on average across these SNPs, are we getting kind of the right thing? The other test that Patrick's going to talk about is the same thing, trying to, use information across many SNPs to say
so
something out of a small sibling sample.
Why not just see if like prediction works similar among between for just the general population and then for siblings?
Yeah, so that's something you could do. It's still a little bit messy because there's I mentioned this I think earlier in the week. There's two kinds of stratification that may exist if you do this prediction thing, right? Because you have your betas, which may be biased due to stratification, but then you're taking these betas and creating a predictor with genotypic data in your prediction sample. Your prediction sample may also be stratified. And so if we then try to do prediction, we were seeing if the within family predictive power is the same as otherwise, right? It's
You want to check for stratification in the discovery sample before you even.
well, this is what we're doing right now. We're trying to see if it's there,
Gotcha, yeah.
right? And so it's true. You could do something like that, but it's a little tricky. There's a lot of stuff happening.
Just to add to what Dan said, I think, if I remember correctly, to replicate the top EA SNP in siblings and within family analysis, I think we calculated at some point you need 40,000 SNP pairs. So that's
yeah.
why, so that's why you can't. You should try to replicate it in a few thousand pairs and it doesn't show up at a 5% level. That in and of itself doesn't tell you anything.
And if you combined every single within family data set that's like, of moderate size, then I'm not even quite sure you get to 40. So
right.
So it's
That'll change with the UKB, but
yeah. Great. Should we talk about the regression test now?
Yes.
Okay. So let's say that instead, so this test just tells us there's a genetic signal here somewhere. But we might want to know how much of the variation is due to true genetic signal and how much of the variation is due to stratification. Because maybe there's just a tiny bit of stratification or maybe there's a lot. And so what I want you to think about is, let's say that we took our within family betas and we regressed them onto our GWAS summary statistics. Okay? So if we did that, I'm going to write this on the board because it'll help, I think, see what's happening here. And it's quite short, so it'll be easy. So I'm going to use this equation that we had before. The coefficient of this regression is going to be the covariance of the within family beta, the estimates there, and the estimates of the GWAS beta, divided by the variance of the GWAS beta. GWAS beta. Okay? So this thing is just equal to what I'm going to call the true signal plus the estimation error. So I'm going to say this thing is equal to covariance of beta plus the within family error. All right? And this one is the true beta plus biases due to stratification plus error of the GWAS. Okay. Now I've made a big assumption. Whoa, this is fun. Okay. So I just made a big assumption here. Can people see what I've done? What did I do here that wasn't just a straight up going from one line to the next? Give you a hint. It has to do with subscripts. And the dog that didn't bark.
You're thinking that the betas are symbols between family and in the general population, which need not be?
Exactly, yeah. So right here in this line, I've assumed that the within family beta is the same as the cross-population beta. And so what we're about to do here is going to rely on that. Maybe when we're done, we can talk about what that actually means exactly. And so the variance of just the GWAS estimate is, I'm just going to write that as the variance of the true betas plus the variance due to stratification plus the variance due to the error in the GWAS. Okay. So assuming that we've done our within family estimates, if our within family sample is independent of our GWAS sample, then the error here is going to be independent of everything over here, so we can ignore it. And same on this side. If the samples don't overlap, then the error in the GWAS is going to be independent of the error in the thin family and the true betas, so we can ignore this too. And then I'm further going to make an assumption that the biases due to stratification is independent of the sample size. That may or may not be reasonable. You could probably come up with a story, but we're going to use that as just kind of an approximation here. So now all we've left with in the top is the covariance of beta with itself. And so that's just the variance of beta over variance of beta plus variance due to stratification plus variance due to error GWAS. Right? So the variance of the error, that's what standard errors are trying to estimate. Right? We want to know how much variation is due to the error. And so we could use our standard errors and subtract this term out of the denominator. So that's something we could do. So I'm just going to cross this out because I'm going to say we're going to subtract that out. So now all we're left with is this. And so you can interpret this as, once we do this regression and then correct it for the error of the GWAS, it's going to be the variance due to genetics over the variance due to genetics and stratification. So it's the fraction of the signal in the betas that's due to true genetic effects and not due to stratification. So if there's no stratification, what is this equal to? It's 1. And so 100% of our signal is genetic signal. But what if this is like 70 million? Then that's going to start going down to 0, right? And so we can think of it really as this fraction of the variation that's due to signal versus stratification. So yeah, so we can do these regressions. But again, let's do we want to talk about this? I feel like I'm way over time already. Should I just keep going?
I think you should keep going.
Okay. We can talk about this like one-on-one later if you want, this assumption here. But we do rely importantly that the effect of the SNP within family is the same as across families.
How accurate is the assumption that siblings, alleles that they inherit from the parents is random? Because aren't there some studies that show that if you're likely to have a child of one sex, then you're more likely. I mean, it's not completely randomly distributed, is it?
So I mean, I guess if parents are making choices and they have children until they have a smart child, then I guess that could. Because I mean, that's effectively what these sibling disparities are, right?
And even if there's sort of imprinting of some sort, would that affect some of these assumptions?
I think it might only affect if there is selective abortion, right? So if there is abortions that you don't observe and you only look at siblings that survives, then it might be true. But
Yeah, so things like that, or even just like the stopping problem I talked about would also lead to differences if parents are choosing whether or not to continue having children based on kind of the genotypes of the children they already have. But my,
Are you sure about that?
if a parent has children until they have a boy,
Yeah, does that, that's not, okay.
and it's 50-50, then you're going to have a lot more boys in the population, right?
You're gonna have 50% boys always, I think. I don't see how it.
Am I thinking? So, okay. Well, I'll have to think about this. Apparently, maybe I'm being dumb. Well, yeah. I don't think these effects. I don't know of people who think about them. I don't know if they're large. So I'm sorry. I don't know.
I think it's fine. What can happen is that you end up drawing a sort of selected sample of SIBs. So it may be that within the population as a whole, the genotype draws that the siblings get are independent. But then you go out, you have to sample a couple of them, they end up with an estimation sampling, which is not true that these things are independent of potential confounding factors.
Mm-hmm.
But that said, if your sampling is not.
Oh, you want to? Okay, we'll do
Sorry?
short.
If your sample is not really large enough.
It's not about size in my example, it's about selectivity and like ascertainment bias or sample selection bias.
Okay.
So you could test that in some way. You could test whether the score predicts whether you're male or female, something like that, which of course it shouldn't because it's based on autosomes only. So anyway.
Yeah.
So given
Oh,
that, like,
we'll go back
in the
here.
second line, the covariance of, and then you get rid of the subscripts. So now we're working with like the true underlying data, which we think is the same, right?
Yeah.
Like, and then you're assuming away the error. It seems like just because, I mean, I'm not sure, right? But like we're ascertaining this through using like very similar SNP chips, and like these things aren't perfect. Like, isn't it kind of believable that like the errors could be correlated between those two?
With the betas?
Yeah.
I mean, yeah. So I mean, I guess if there were, but like you would need the error to be correlated with this beta.
Okay, it's not a problem with the errors.
And so like what we're relying on when we assume that this is independent of that is that the samples are different. And so if biases are getting introduced, over here due to stuff like that, I wouldn't necessarily think it would be correlated with the ones over here, unless like have the same kinds of things in both, I guess. But
So there's one huge way in which those two betas are different. The typical GWAS one includes the effect of parental, the gene on the parental nurture, and the one that's within family doesn't. And so the betas from the GWASs are going to be enhanced for anything where there is kind of nurture transmission from parent to child relative to the within family ones that don't have that enhancement.
yeah.
So that'll typically lead them in the same direction, but you're going to have this enhancement for the things that have nurture transmission from parent to child as well as a genetic effect operating on both.
Yeah. So that's one example of why beta within family might be different than beta across families. You could also have another thing if you have like these super egalitarian parents that make it so that all their kids are exactly the same phenotypically, then that could mean, what would that do to the within family betas? So let's say that within family there's no variation in the phenotype because the parents are forcing it. What is that going to mean for the within family beta? It's going to be 0 because there's no within family, right? So there's going to be variation in the genes, but there's going to be no variation in the phenotype. Or if you had an example of a family where they perhaps have limited resources, and so they pick the child who they think's the smartest one, and they sink all of their educational resources into that child, what's that going to do to the betas? Yeah, it would in principle make it bigger because they're kind of exacerbating these genetic effects. So I mean, there's lots of reasons why you maybe wouldn't believe this, and there's some, and in the EA3 we're seeing some evidence that might suggest that that these differences are non-trivial, and we're still trying to wrap our, we're working on details on that, but. Okay. Great. So I don't know. Should I just keep? Do you want to take a break, or do we want to? How long have I been going? I don't even know. Are people exhausted? I can just keep going. I'll just keep going, unless you guys. This is the really fun stuff, anyways. You're gonna love this. Okay. So now we can see. We know if there's stratification in our data or not, right? And so we see that like, oh, our data's stratified, this is bad. What can we do to fix it? And so we know it's here. We want to talk about things that we can do. And so there are some approaches that work by adjusting the standard error, and there's some approaches that work by adjusting the effect sizes. And so I'm going to talk about those. So let's talk a little about what standard errors are supposed to represent. So we have these things as standard errors, and they're telling us like, oh, is this significant or not? But we want to think, what are they supposed to mean? And I think one reasonable way to think about what a standard error is it's supposed to be a measure of how much variation there is in our estimate due to stuff that we don't care about. And so most of the time, the stuff we don't care about is sampling variation. Another thing that we think we maybe don't care about, though, is biases due to stratification. And so this is kind of the rationale behind these types of methods that adjust the standard errors. We're saying, okay, well, currently our standard errors are just talking about variation due to sampling variation. Maybe we can make them a little bigger and also capture variation due to these biases. And so the classic thing that had been done for a long time, you saw in a lot of papers until just a couple years ago, was something called genomic control. And so what they did is they took they did their GWAS, and they listed them in order based on how significant they were, ordered by the chi-squared statistic. So you might be really familiar with what like the t statistic or the z statistic is. It's the beta over the standard error. The chi-squared statistic is just that thing squared. And so this way, with z's, you're looking for the absolute value of z, but in chi-squared, it's just bigger is stronger. And so so they take the chi-squared statistics, and they find the median chi-squared statistic. Now, under the assumption that fewer than half of the SNPs are causal, that means the median SNP should correspond to a null SNP. So if half the SNPs don't have any effect, the one right in the middle, in principle, should be one that doesn't have any effect. And so you can look at the chi-squared statistic of that. So a chi-squared statistic has mean 1. And so you might look at that and you see that the value of that chi-squared statistic there is like 1.3. And so that 0.3, under the assumption that this median SNP is actually null, the 0.3 we're saying is how much variation there is due to stratification, because the only thing that we think that the SNP is stratification. Because the SNPs that are also include genetic effects are above the median. And so we inflate our standard errors by the chi-squared of the median SNP divided by what we expect the chi-squared of the median SNP to be, and that should kind of give us a sense of what the right how much we should be inflating things. But this is a problem, right? So we've made this assumption that half of SNPs are null. And so in what settings might that be reasonable? When do we think that doing genomic control might be the right thing?
Extremely polygenic, well, if it's not extremely polygenic, perhaps.
Yeah. So it's these Mendelian traits that Fleur was talking about. So if there really is only one, like we think that there's only one SNP or maybe a small number of SNPs. So a lot of medical traits are like that, and that's why this originally was adopted in the medical field, and so that maybe was reasonable in that case. For our complex traits, though, it is probably a really bad assumption, and this is going to be really bad for us. Because let's say that the median SNP actually has some genetic signal. What's that going to mean for the chi-squared statistic of that SNP? It's going to be bigger because it's more significant because it's capturing genetic signal. And so when we divide by that, we're going to divide by a bigger number, and we're going to be canceling out a lot of genetic signal. We're losing a lot of power by doing this genomic control. So we think that we're just inflating based on how much variation is due to stratification, but we're also inflating based on some of the genetic signal. So I'm just going to show you some pictures. So these are QQ plots, and what we do here is we on the x-axis here, this is the expected p-value under the assumption that every SNP is null. So if every SNP in the genome is null, p-values should be uniformly distributed. And then over here we plot the observed p-value. And so under the null, if the p's are uniformly distributed, we'd expect this to go right along the diagonal. But deviations from the null like this means that we're observing p-values bigger than what we expect. And so what we're looking for when we look at a QQ plot like this is deviation away from this line. Now, if the assumption of so let's talk about things that will look different. If there's no stratification, but only a small fraction of SNPs are causal, then what will this look like? Well, our hypothesis is holding all the way up here, so it's going to follow the line really closely. But then once we start hitting causal SNPs, this is going to peel away there. If there's stratification, though, and so every SNP is kind of associated due to these stratification things we don't like, it's going to start peeling away early. But that's also what's going to happen with polygenicity. And so I have some traits here. This is tuberculosis, and it's peeling away early. We have education here. It's also peeling away a lot earlier. Height peels away even earlier than that. And so, but for these traits that we think are polygenic, right, I'm doing a bad job explaining what this is, but we can't differentiate between the two. And even traits that we think are highly polygenic, like height or education, look the same. I mean, I have no idea what the genetic architecture of tuberculosis is, but I tried to pick one that was medical and maybe was slightly more monogenic. But so this is just to highlight what the problem is with genomic control in the settings that we're interested in.
What's happening with the standard errors? What are the standard errors on the 45 degree?
So this is just based on, so there's going to be so under the null, the p-values are uniformly distributed, but there's some just noise due to sampling variation.
Why is it increasing as you increase?
Because under the null, there are very, very few p-values. You should expect very, very small number of p-values that are
Yeah, yeah. Okay, great. As an additional piece of evidence of how things are really polygenic, this is kind of a fun picture that I like. So what they've done is on the x-axis here, this is the length of the chromosome. So like this point corresponds to chromosome 7, and it has length 160 or something. And then over here, this is the fraction of the heritability described by that chromosome. And it looks like the relationship between how much heritability the SNP explains is proportional to its length, which kind of signals that the signal for height is spread throughout the whole genome. And so this might also make us think that it's polygenic and genomic control might not be a good thing. LD score regression also allows us to do a similar thing, but more principled, and Raymond will talk about that tomorrow too.